Okay, so we finished up last week the session on the series of lectures on replication, DNA replication, and now we're going to transition over the next 3 lectures talking about both translation today. So what are the basic mechanisms of translation? How does that work? On Thursday, we'll talk about regulation of transcription. So we're not going to talk about any regulatory control today, just how it gets started, how it processes, and how it terminates, and what happens to the mRNA. And then next Tuesday, we will have a final lecture in this quarter, which is on translation. So how do we get from the mRNA structure into protein? Okay? All right. So we're going to couch the series. Wow, that's really loud. Turn that down. Okay, so we're going to couch the lecture on transcription in the confines of diabetes, as well as the use of anabolic steroids, something that plagued Major League Baseball in the early to mid-90s. All right, so let's just really briefly cover what diabetes or diabetes mellitus is. Actually, I know this pretty well. My father was a physician, and his specialty was diabetes. So we have a diabetes day camp every single year when I was a little kid. And so whether I wanted to or not, I got to learn a lot about this disease. So it's a group of metabolic diseases. You guys will talk about metabolism later, actually almost at the very end of the term. But we'll go ahead and introduce metabolic diseases here. In particular, though, we're going to focus on an aspect of diabetes which has a defect, where a population of patients have a defect in their translation. All right, so diabetes mellitus is a group of metabolic diseases basically characterized by this hyperglycemia, so an elevated glucose level in your blood, and a chronic hyperglycemia. Okay? And this is going to result from either defects in insulin secretion. We'll talk about why that might be. Insulin action, so its ability to carry out its function, or in some cases, dysfunction in both of those. So can anybody in the room tell me what insulin does? Any ideas? Okay, yes. Yeah. So any more details on that? How does it do that? Yeah, that's exactly right. So how insulin does it is it facilitates the recruitment of a glucose transporter. So remember, glucose is a polar molecule, cannot traverse across our plasma membrane, requires a channel, a glucose transporter in this case, in order to allow it to pass the plasma membrane. And so insulin is essentially regulating the localization of that glucose transporter to the plasma membrane. So when we knock that out, now you wind up having cells bathed in nutrients and can't actually utilize them because they can't get it into the cell. Okay? All right. So chronic hyperglycemia diabetes is associated with a lot of different long-term damage, dysfunction, and failure of organs, in particular the eyes, the kidneys, and we'll talk about those too, but also nerves, heart, and blood vessels. And primarily, hyperglycemia is going to impact your small vessels, so your vascular system, your peripheral vascular system. And so a lot of the tissue damage and destruction and inability to repair is a function of poor blood flow in your peripheral systems. Okay? All right, so some of the symptoms are polyuria, which is basically expelling the frequency of urination. So patients need to urinate significantly and frequently. And this is a function of high glucose levels getting transported across the glomerular network, right? And acting as an osmotic pump, basically, to pull fluids out of your bloodstream and into your kidneys. And so as a consequence, patients will have to urinate a lot. As you can imagine, if I start peeing a lot out, now I'm going to need to drink a lot. So I have this need to drink a lot of fluids. My mouse just keeps doing this, okay? All right. Interestingly, you have abundant levels of glucose in your bloodstream and surrounding cells, and yet patients will have a limited amount of weight loss, certainly impaired growth of cells. Again, I mentioned this idea of cells starving in a sea of nutrients, in particular because they can't get this across. So when cells have difficulty accessing glucose and undergoing normal metabolism, they'll engage other kinds of mechanisms, other kinds of forms of respiration, cellular respiration. And one of those will generate ketone acids. And so patients with diabetes have this ketoacidosis, which is a buildup of acids that are based on the breakdown of fats. So fats are our long-term storage of energy, and cells will begin to engage that. As a consequence of all of these, diabetics tend to have sweet-tasting sweat because the glucose gets into their sweat. They also have acidic blood, which, as you can imagine, is not great. All right, so the long-term complications are, again, primarily associated with loss of blood flow, loss of efficient blood flow in the peripheral systems. Patients will develop retinopathy, so a loss of retinal function due to, again, low blood flow. Some diabetic neuropathies, so your nervous system, especially your peripheral nervous system, needs a significant amount of oxygen and nutrients. And if you start getting low blood flow in your peripherals, like your fingertips or your feet, then your nerves will wind up becoming less effective. Interestingly, this has a lot to do with also diabetic ulcers. So again, patients will wind up losing nerve sensation and developing things like bed sores, and then they don't even know that they have an injury because their nerves are deadened in that area. Because they have poor blood flow, then they also can't heal that wound. So they get these ulcerations that persist over time, and it's a pretty big problem. Okay. All right. Cerebrovascular, again, due to loss of blood flow up in your brain. Hypertension and abnormalities with lipoprotein metabolism. Periodontal disease, again, a function of poor vascular flow. All right, prevalence and the economic impact of diabetes. You guys probably are fairly familiar with this. It's a huge healthcare burden in the United States, actually in most developing worlds or developed worlds. In the US, we're talking about, and these numbers are a little bit old, but about $250 billion in total cost. About 2 million people per year in the United States are afflicted with diabetes. And interestingly, the traditional cultures of developing nations are actually, there's not a huge prevalence, so it's fairly rare. So this is, in fact, a disease of development, essentially. And as we will talk about here in a minute, a lot of it has to do with diet. Okay. All right, so it does vary by race in the U.S. general population. And interestingly, there are some Native American populations that have what appear to be a genetic predisposition to this disease. And so there's ongoing research to understand what those key genetic signatures are that predispose these folks to diabetes. All right, so when we talk about the etiology of the disease, we really have to now subtype diabetes and we're going to subtype it into 3. Officially 2, but we're going to talk about a third. Type 1 diabetes, also known as juvenile diabetes. These are patients that have inherently within them either a depletion of the beta cells, which are the insulin secreting cells in your body, or, and I believe Dr. James talked about autoimmunity with you guys, you can also have an autoimmune reaction against your own beta cells, and you can actually significantly wipe out the beta cells in the pancreas. Now, if I can't make insulin, then obviously I have a difficult time locating that glucose transporter to the cell membrane. The other type is type 2, or adult onset diabetes. This is the one that's way more prevalent in developed countries. It has to do with results from improper regulation of insulin or insulin signaling. You'll notice that oftentimes we equate diabetes, the prevalence of diabetes, to obesity, and they are linked in a sense that if I have a huge glucose burden, chronic glucose burden due to perhaps a dietary concern, then I can lose my insulin sensitivity, right? And so we're losing the sensitivity of those beta cells to sense how much glucose you have in your bloodstream and produce insulin in kind. And so there's a lot of work on engineering beta cells that have a retuned sensitivity to glucose, injecting them back into the pancreas or into the peritoneal cavity to try to get a sort of pseudo-pancreatic organoid tissue growing. And so there's a lot of effort in there. But what we're going to talk about, well, that was interesting. What we're going to talk about is this oftentimes called type 1B or 1.5, type 1.5 diabetes. It's really a type 2 diabetes known as mature onset diabetes of the young. And this is a function of a transcriptional defect resulting from an inherited mutation. And so this is we will couch our discussion of transcription and transcriptional regulation in the confines of this disease, MODY. All right, so let's move away from the clinical descriptions and get into what transcription is really about. And similar to DNA replication, we're going to take let's talk about it in prokaryotics, which is a simplified system, and then we're going to compare and contrast that with the eukaryotic system, which tends to have much more regulatory control, much more complexity. Okay? All right, so when we talk about transcription, we're really talking about 3 primary stages. The first is that of initiation. The second is elongation, and the final is termination. Pretty easy. We've got to initiate a process, we've got to elongate a message RNA, an RNA strand, and then we have to terminate the process, right? So that should be fairly straightforward to think about. And it's also very similar then to DNA replication, right? We had these origins of replication, which were initiators, right? We had initiating complexes that started the process. And then we had termination processes as well. So when we talk about transcription, in eukaryotes, we're really talking about single genes, okay? So we'll talk about this thing known as I'm sorry, I keep messing with my mouse. I'm trying to get it to go live. We're going to talk about this transcriptional unit. And in eukaryotes, that will be one gene only. In prokaryotes, it can be many genes, okay? It can be one gene or many genes in prokaryotes. All right, so the transcriptional unit is pretty straightforward. We're going to have this promoter region, which is sort of analogous to that origin of replication, right? It's where transcription will be initiated and will begin. We then have a coding region, which is here in the middle, and then we're going to have some kind of terminator sequence. All right, so this is a really basic overview of it. We are going to have, essentially, when you have this transcriptional unit, you have an RNA polymerase coming in, and I'm going to add one thing to this, and I will highlight it again because I want to add it into this lecture. You're going to have, in the case of a prokaryote, you're going to have RNase, and it's going to be facilitated by a protein known as sigma factor, okay? And I will add that to the slides, and I will modify them when I put them, and I'll modify them online. So what sigma factor is sort of analogous to those initiator proteins in DNA replication. So it is a factor that's going to recognize, and we'll talk about it here in a minute, other consensus sequences, which help facilitate RNA polymerase to bind that area, okay? All right, so our initiator in the case of transcription in prokaryotes is this factor known as sigma factor. It's a protein known as sigma factor that helps recruit RNA polymerase to that promoter region. It's going to partially unwind the DNA, and then it's going to proceed in copying what we call a template strand, copying that template strand, or using that template strand to copy the DNA. So instead of having, again, sort of the compare and contrast with DNA replication, instead of having a parent strand and a daughter strand, we've got different terminology here. We've got what's known as a coding strand here, which is, in this case, going in the 5' to 3' direction, and a template strand. And we'll talk a little bit more about that terminology here in just a minute. So the RNA polymerase is going to unwind and extend, right? It's going to polymerize RNA using that template strand. It's going to proceed along that strand until it gets to this termination sequence. And then it's going to fall off, and then we're going to have this RNA transcript. So that's a very high-level view of it, and let's dig into some of the details. Okay, so what do we mean by template strand versus a coding strand? Let's get some of the terminology down. And it's a little bit different. Template strand, for you guys, might be fairly intuitive. Template strand is the template that the mRNA polymerase is going to use in order to undergo, right, to facilitate complementary base pairing and polymerization, right, the formation of those phosphodiester bonds, which is going to string a polyribonucleic acid, right, the RNA. All right, coding strand, if you think about it, is also obvious, but it might not be immediately obvious, because, so if we take this example down here, and we'll talk about all these other details here in a minute, if the RNA is, let's say, starting at this point, at the +1, the initial position, and transcribing off of this template strand, it's transcribing a reverse complement sequence, right? We talked about reverse complement sequence and complementary sequences earlier ago, right? So it is synthesizing using the strand from the 3' to the 5' direction to synthesize a ribonucleic acid in the 5' to 3' area using complementary base pairing, right? So if my template strand has a T, then my mRNA has an A, right? If I have a C, then I have a G, right? And so what the coding strand does, but that's the exact same for the coding strand, right? So you can think about the coding strand as actually giving you the direct sequence of what that messenger RNA is going to be. Does that make sense? So we have basically two complementary strands to that template strand. One is the DNA coding strand, and one is the mRNA strand. And they're identical except for what? What were you going to say? Yep, that's right. Wherever the DNA has a T, the RNA has a U. Correct. Okay? So the coding strand, if I tell you, here's the sequence of the coding strand, you should immediately know, that's the sequence. That's the exact same sequence as the mRNA. Okay? The template strand, you have to do that conversion of writing down the complementary sequence. Correct? Everybody understand that? Okay. All right. So coding gives you the code, basically. All right. Terminology. Upstream. So there's always a starting point to transcription, and we'll talk a little bit about what that transcriptional start sequence is here in a little bit. When we talk about upstream sequences, all of the terminology is associated with the coding strand. Okay? You can think about the template as only really providing the copy, right? The complement sequence. So when we talk about upstream direction, we're talking about prior to or before the start point. which would be position 1. So you can see these would be, sometimes you'll see these designated as X number of base pairs upstream, or you'll also see them sometimes designated as the minus 30 to 35 sequence, which means it's, or 35 to 30, right? So it's like T minus 10, 9, 8, 7, so they're counting down until they get to that transcriptional start, and then it goes up in sequence, 1, 2, 3, 4, 5. Does that make sense? Okay. All right. So upstream in the 5' direction on the coding strand, that's up here. And what we're going to have is this, this sequence that's known as a promoter region. And the promoter region is going to be where the RNA polymerase is going to bind and going to unwind the DNA and begin to transcribe. And it's going to use a terminology, something that we've talked about before, which are consensus sequences. Remember, we talked about consensus sequences in prokaryotic DNA replication as these repeated units that define where the origin of replication is, because they're proteins that recognize those specific sequences no matter where they are in that bacterial genome. Okay? The same exists here. Yes? No, no. So sigma factor is going to recognize consensus sequences that are indicative of where transcriptional where RNA polymerase needs to bind, right? The factors, and now my brain is, I've already forgotten the, or cannot recall the initiator for the origin of replication in prokaryotes. It's going to recognize the specific origin of replication in that circular DNA, right? They're two separate processes. So consensus sequence, remember, is a general term that is a sequence of DNA that has a conserved function, okay? So consensus sequence, when we're talking about DNA replication, is a conserved sequence across oftentimes many prokaryotes that identifies where DNA replication will begin. And there's specific proteins that recognize and bind that region. These are consensus sequences that are driving the recruitment of RNA polymerase to genes, right, to be transcribed into protein. Okay? Okay. All right. So again, it's a great opportunity to say that consensus sequences, remember, are these sort of alignments where we have consensus, meaning similarity or even equivalency in gene sequences across multiple species that are triggering conserved function. In the case of sigma factor, transcription. Okay? All right. Okay. So what's happening at initiation? Let's get the process started. So I told you that sigma factor will bind those consensus sequences and facilitate the recruitment, enable the recruitment of RNA polymerase, the recruitment and binding of RNA polymerase also to those consensus sequences that are within the promoter region, right? So you can think about promoter regions as promoting gene transcription, okay? All right, so the RNA polymerase is going to bind with the help of sigma factor. RNA polymerase will melt the DNA, so it's capable of doing that itself. And it's going to stall. It's going to kind of hang out for just a small little bit on that promoter region. And what it's doing during that little stalling event is it's beginning to synthesize the initial polyribosomal nucleic acid. Okay, so it's going to add those bases one by one until it has about 9 bases. Once 9 bases have been added, then it's going to start trucking down the DNA strand. Okay? All right, so meaning RNA polymerase can move. Okay? So the movement, again, it's reading, and this is, again, it's very easy to get your directionalities messed up. I warned you on the DNA replication side of things, it's even worse for RNA polymerase. Remember, it is writing, quote unquote, writing a 5' to 3', so it's polymerizing in the 5' to 3' direction. So it's actually using the template DNA in the 3' to 5' direction, right? Because it is literally transcribing the reverse complement of that template strand. Okay? All right. So RNA polymerase, unlike DNA polymerase, it can actually unwind DNA. It unwinds the DNA. It synthesizes this polyribonucleic acid. And let's see, we'll go a little bit further. So it can unwind the DNA. It'll add those ribonucleic acids, right? Those NTPs. And we're going to have about, in that RNA polymerase, about 12 base pairings between RNA and DNA. So this is this RNA-DNA hybrid region. And there are about 12 base pairs in that region before they peel off, before that RNA peels off from the DNA. Oops. All right? And the DNA is then going to rewind. It's going to be rewound. Rewind sounds weird. It's going to be rewound on the back end of that RNA polymerase. Yes? Yeah, good thinking. The answer is no. So the difference between DNA replication is, you can think about that rope diagram that the video came up for. When you begin to pull and pull and pull, those things never get rewound on the back end, right? This is more akin to having a wound rope, shoving your hand in the middle of it and sliding it back and forth, right? So there's not that supercoiling effect because we're only unwinding a very small portion of the DNA and then it's rewound. Does that make sense? Okay. All right. Again, RNA polymerase is going to elongate from the 5' to 3' end, and thus RNA polymerase moves along the template strand from the 3' to the 5'. Yes? Yes, there are, and we're going to get to those. Yep, absolutely. Okay. All right. So are there any questions about elongation? It's the difference between RNA polymerase and DNA polymerase? Yes?
So is the RNA strand moving left
So you asked, you guys can help me remember to repeat the question as well. It would help, because I know you guys can't hear it. So you're asking, is RNA polymerase moving to the left or the right? In this case, it's moving to the right. Okay? So you can see the light blue strand is the 3' to 5' direction, right? And so it's polymerizing from 5' to 3', so it's using that strand. That was a bad description, but yes, it's moving to the right. Okay? All right. Okay, so let's talk a little bit about termination. So termination is really defined by the secondary structure of the transcript. Similar to proteins, RNA can actually fold on itself and form complementary base pairing within a single strand of RNA or even DNA, which can cause problems, right? In this case, what happens is towards the end, at the end of the DNA sequence where, let's say the important stuff, well, the important stuff is all upstream, up here. When we get to the end of the transcriptional sequence, we're going to have these self-complementary sequences, basically repeats. They're palindromes of each other, right? So that when they're transcribed, then they form complementary base pairing, and you can get this structure known as a hairpin, right? So the hairpin is determined by this little linker has to be a sufficient length so that we can get bending of that RNA, and this sequence and this sequence have to be complementary for one another. And so they'll form. These happen to be, tend to be GC rich. Can anyone give me a reason why they might be GC rich? Yeah. Correct. That's exactly right. So there's more hydrogen bonds between G's and C's, and therefore the structure is much more stable. It's also, there's a lot of tension. So unless that loop structure is huge, there's a lot of tension on that hairpin, and you need that hydrogen bonding to hold that closed. There's some really cool RNA probe technology based on this kind of stuff. All right, so the RNA polymerase is going to transcribe these complementary sequences. We're going to get this structure, this hairpin loop structure that forms, and one of two mechanisms will happen. In some RNA polymerases, the RNA polymerase itself has a recognition site for that hairpin. And when it recognizes that hairpin, it turns that RNA polymerase off. In most cases, the hairpin is recognized by accessory proteins that then would exhibit allosteric control over that RNA polymerase and turn it off. Okay? All right. All right, so as you can imagine, so here's our RNA molecule, 5'. We're trucking down 3'. Here's the important region up here. We generate this termination sequence. And because it requires sufficient time for this complex to recognize and form on that hairpin, the RNA polymerase is just on, right? It's got an on switch, it's got an off switch, and it is on. And so it just keeps on trucking. And so oftentimes what you'll find is that, well, almost in every single case, the RNA strand that's generated is far longer than the really important region, right? And so it's going to keep on trucking as long as it's on until this is recognized and it's turned off. Okay? So we're going to now talk a little bit about the eukaryotic system, and then we're going to talk a little bit about how we deal with some of these extra bits and pieces on the RNA strand. So contrary to prokaryotes, eukaryotes have a number of different polymerases, and we're going to talk about 3 of those. One for each major type of RNA. We're only going to focus on this lecture on RNA polymerase II. Okay? But for completeness sake, we'll talk. I'll mention that RNA polymerase I makes pre-ribosomal RNA. That's the little R, stands for ribosomal RNA. And that'll be a part of that ribosomal complex that helps us read the mRNA and turn it into protein. RNA polymerase III makes what are known as transfer RNA. So those are RNA structures that have amino acids tethered to them, recognize the codons, and carry the amino acid that correlates to that, that trimer, that 3-mer codon sequence. Okay? RNA polymerase II makes pre-mRNA. So I want to highlight that all of these are termed pre, and that's because in every single case there's processing that occurs. So this would be the immature form of each of these RNAs. Okay? These 3 different classes of RNAs. So each polymerase uses a different promoter structure. So there's certain promoter structures for ribosomal RNAs versus tRNAs versus message RNA. So that's the mRNA part. Okay, so let's talk about the RNA2 promoter because we're talking about transcription here. There is, again, an initiator sequence surrounding that start point, so similar to the prokaryotic system. And there's a very specific start point. It is this APY, and the PY is the pyrophosphates, so it's going to be C's and, no, what's my brain thinking? C's or T's. So it's the pyrimidine structures. So APY5, upstream is a PY2 followed by a C. So this is a very specific starter sequence. All of your start sequences, we'll get into this in the translation component, they all code for a methionine. So you can almost always tell this, you can always tell the start of a protein sequence, where the protein's going to start, because it codes for that first methionine, which is subsequently cleaved off. That's for another story. So there's a lot of detail here. Some of it I'm going to breeze over for the sake of your sanity. and others I'll delve into. Obviously, you're responsible for what I dig into and not what I don't. All right, so how do we determine that initiator sequence? It is, again, another consensus sequence known as a TATA box. Okay. So the TATA box is somewhere around minus 125. Right? So remember that, that designation, minus 25 is 25 bases upstream in the 5' direction of the coding strand of that DNA. Okay? So it's not exactly 25, but it's around kind of in that zone. All right? And it's the reason it's called a TATA box is it has a sequence TA, TA, sometimes TA. Okay? All right. So this is going to designate the initiating spot or initiating sequence. And so you ask, is there a correlate, right, to prokaryotic sigma factor, and that is known as transcription factor in the eukaryotes. Now, transcription factor or transcription factors, plural, it designates an absolutely enormous class of regulators of gene transcription. We're going to talk about some today that are the critical ones for actually initiating the process of RNA polymerase, right, or formation of mRNA. In the next lecture, we'll talk about a lot of the regulatory transcription factors. So again, in the eukaryotic system, there's a ton of complexity in how we regulate what gets turned on and what gets turned off, okay? So transcription factor, very vague term there, binds to the TATA box and helps facilitate recruitment of RNA polymerase II, similar to the prokaryotic system. So, so which transcription factors exist in eukaryotes? We're going to focus on today's lecture on this one transcription factor known as the basal transcription factor. Basal transcription factor is this TF2D molecule, okay? So TF2D is always required to allow RNA polymerase to bind to DNA. That would be different than a lot of the regulatory transcription factors that we're going to talk about on Thursday that in various combinations can influence the accessibility of that TATA box, right? So in influencing the accessibility of the TATA box, we're essentially regulating the probability of that gene being transcribed, okay? The basal transcription factor is absolutely fundamentally required to initiate transcription. All right, so let's go through this in a cartoon way. So the TATA box somehow becomes exposed. We'll talk about that regulation on Thursday. TF2D binds to the TATA box. We then have accessory proteins, that will form a pre-initiation complex. Again, we're not going to harp on what those proteins are. They're complicated, and that complex gets very big. It forms this pre-initiation complex, which then is going to recruit RNA polymerase to that initiation site. The little black S is for the start base. So it's an A, usually followed by a T and a G, but can be variations of that. And then RNA polymerase is going to proceed to transcribe that gene sequence. Okay. Are there any questions about that before I move to what we're going to do with that mRNA after it's been synthesized? Yeah.
What's the difference between the regulatory transcription factor as compared to the basal transcription factor?
Yeah, so I gave you a little bit of a sneak peek into Thursday, and maybe I shouldn't have, but regulatory transcription factors, which we're going to talk about on Thursday, are essentially, they're regulating the accessibility of that TATA box. When that TATA box becomes exposed and accessible to TF2D, it will bind. I can have, if I deplete a cell of all of its basal transcription factor and I activate other transcription factors to open up that TATA box, I still will not get transcription, okay? So this is the one transcription factor that really initiates the recruitment of RNA polymerase and gets the process started. There are things that happen upstream of that which regulate whether that TATA box is hidden or exposed, okay? And we'll talk about that on Thursday. All right. Any other questions? Okay. All right, so we've made a primary transcript. That will be a pre-mRNA. And now we're going to try to make that pre-mRNA into a mature mRNA. And three things are going to happen to that mRNA in eukaryotes. One is termed the 5' cap. And just as the term suggests, we're going to cap that 5' end. The second thing that's going to happen is the formation of what we call a poly-A tail. And that'll happen on the 3' end. And then the final thing that will happen is we'll splice that mRNA. Why would, as the name implies, why would we want splicing? Yeah. To get rid of the introns. That's exactly right? So prokaryotes don't have introns. We do. And so it's a way for us to begin to mix and match our exons as well. So there's really interesting studies on regulation of why splicing occurs in certain regions. It's a whole field known as alternative splicing that derive from a single gene, multiple protein species from even just a single gene. So I'll talk about that here in a minute. All right, so one of the first, so I will mention that the 5' cap and the poly-A tail absolutely fundamentally come before splicing. So no splicing will happen until those two modifications are made. Yeah.
So would you say that 5' cap poly-tail are prokaryotes as well as the splicing we have with the 5' poly prokaryotes and eukaryotes? Splicing for eukaryotes?
No, I would not say that. This is a primarily eukaryotic modification to messenger RNA. And it has to do explicitly with the anatomy of the cell. The difference between the anatomy of a cell, specifically the creation where we evolved a nucleus, right? So bacterial DNA is not separated from their cytoplasm. Ours is by a membrane barrier. So many of these modifications, well, at least one of these modifications, is absolutely required in order to help facilitate, get the messenger RNA where it needs to be. Speaking of which, that's the 5' cap. Okay, so so one of the first modifications is going to be this capping of the 5' end with this methylated guanine structure. So you can see it here. This is guanosine triphosphate. It has a methyl group here in position 7, and that's what gives it its name. It is, we put it in quotes, it's a backwards cap. Why is it a backwards cap? Well, if you think about a ribonucleic acid, polyribonucleic acid, right, the connections are between the 5' carbon and, the 3' carbon and the 5' carbon, right, as it processes. In the case of the 5' cap, we have the exposed 5' carbon on that very end, 5' end of the RNA, now forming a bond with the 5' carbon of this 7-methylguanosine. So it's a characteristically different bond. It's still a phosphodiester bond, but it's a characteristically different structure than what you would see in the ribonucleic acid, polyribonucleic acid backbone. Okay, and so this gives it sort of a unique chemical characteristic. All right, this is added during elongation. So you've got, as soon as it comes off, you're going to have these capping enzymes that recognize the 5' end, and they form this chemical bond between 7-methylguanosine and the 5' end. So why? So the capping enzymes are only associating with RNA polymerase II, and we'll talk about that here in a minute. The functions of this 5' end are, as I kind of alluded to before, it flags it for nuclear transport. So we have a very complex nuclear pore structure that facilitates the transport of molecules in and out of the nucleus. And the 5' cap facilitates the binding of RNA into to proteins within that pore structure and facilitate its translocation into the cytoplasm. And it's only in the cytoplasm where it'll be transcribed. Okay? The other is it protects it against degradation. So just like we talked about exonucleases, DNA exonucleases that chip at the ends, right? Cleave off the little end nucleic acids. We have exoribonucleases, which are going to do the exact same thing to RNA. RNA is also significantly more sensitive both to enzymatic degradation as well as hydrolysis. And so we want to cap those ends to try to protect it. I'm lousy at dealing with RNA. Some people have a knack for it. When I purify it, it always degrades like almost immediately. I don't know why, but it doesn't like me. I don't know. So we want to protect those ends from degradation. I have a colleague that is bordering on genius at Georgia Tech who actually will further modify engineered mRNAs with different kinds of caps that stabilize them for even in vivo delivery. So you can deliver them in the bloodstream and they stay stable in bloodstream for basically genetic engineering type of approaches, which is kind of cool. All right, and finally, the 5' end is going to be the site that is recognized by the ribosome in order to kick off translation. Okay? All right, so I mentioned that the capping enzyme is only going to associate with RNA polymerase II. Can anyone think of why it might behoove us to have this selectivity? Yeah. Correct. Yeah. So we don't want capping of those different classes of RNA molecules. So ribosomal RNAs we don't want capped, and tRNAs we don't want capped. It would disrupt their function. Okay. All right, so let's talk about the other end. So we've capped the 5' end and we've quote unquote protected it, and that's our signal sequence to get it out of the nucleus, start translation. On the back end, I made the comment that transcripts are generally too long, right? That RNA polymerase is just trucking along. It takes some time for that hairpin structure to be recognized by those accessory proteins and them to exhibit allosteric control over RNA polymerase and shut it down. And so RNA polymerase is still trucking down that gene. And so what's going to happen in the case of the 3' end is we're going to have this protein known as polypolymerase, poly-A polymerase, or PAP, and it's going to find another consensus sequence that is AAU-AAA. Here it is. So there's the 5' cap way on that end. Here's the whole transcript in that little sliver, and now we're looking at the end. So poly-A polymerase is going to recognize that motif, and it's going to cleave the transcript about somewhere between 10 and 35 nucleotides downstream. So downstream is in the 3' direction. So you can think about it like I always think about it like a river. You know, if the synthesis is going 5' to 3', then everything upstream of 5', right, versus, or before 5' versus after. So downstream of this sequence, it will cleave, and then it's going to use adenosine triphosphate, right? It's going to catalyze that reaction of formation of a poly-A tail. Okay, so it's going to synthesize this poly-A structure. Similarly to the 5' cap, the poly-A tail helps protect the transcript from degradation, and we'll talk about why here in just half a minute. And it is also necessary for the full initiation of translation. Yes?
So when you have replication, you said
Yeah, that's a great, yeah, you're thinking about it. It has both indoribonuclease activity, right, because it's cutting in the middle, and it also has polymerase activity, right? So it's a protein with two activities, actually three, because it also has some recognition domain that's binding to that consensus sequence, okay? But yeah, you're definitely thinking about along the right lines. Okay, so how does this work? Let's schematize it here. So here's our 5' end, right? This is our transcript, our mRNA transcript. We're going to generate that 5' cap, so that's the G here. We're going to form that poly A tail, and what's going to happen to the poly A tail is we have a class of proteins known as poly A binding proteins, right? Wildly inventive names. They're going to bind to that poly A tail and effectively protect it. They're both going to protect it from enzymes that would want to bind to that end and start clipping away at it. They're also going to facilitate the circularization of that mRNA, okay? And so what you can imagine is if I circularize the mRNA, here's the start sequence, and I have a ribosome that's trucking along and translating it, and when it gets to the end, it's right here proximal to the front end. And so you can get multiple rounds of ribosomes on a single mRNA. In fact, there are images where they're literally just lined up one after another, packed on a single message, generating protein off of that. Okay? All right. So when we think about mRNA degradation, can anyone give me a reason why it would be important for RNA to be degraded? Right? Why would we ever want it to be degraded? Yeah.
Help recycle nucleotides in the cell.
Okay. If I were to tell you that ribonucleotides are not a scarce reagent, right, or resource, that we have a plethora of them. What do you think? Yeah. So, and you were going to add something, or is that what you were going to say? Yeah. So, I mean, it depends on how much control you want, right? Let's say I'm at a 2-cell stage. Let's make it simple, and one of those cells triggers gene transcription of protein A. If I can't turn it off, then that cell forever and ever and ever is going to produce protein A, right? If that protein needs to be has a very time-sensitive function, then I've lost that ability to regulate that protein over time, okay? And so what we want is it's sort of this yin and yang of making RNAs and them degrading, right? And it's this nice balancing act between how fast we can make mRNAs that we want to make protein and how fast they're being degraded. And the cell has various mechanisms to both facilitate more RNA being produced, and they also have a host of mechanisms that we're not even going to come close to touching, actually we're not going to touch on, that regulate mRNA stability. Either destabilize it so it gets degraded faster, or stabilize it so that it hangs around for a really long time. There's a really cool process in your cells when they get stressed of RNAs getting packaged into these things called stress granules or P-bodies. And the RNAs can reside in those things completely protected from enzymes in these little clusters for days, weeks, months. And when the cell becomes less stressed and those stress signals are gone, then those mRNAs are released, and now the mRNA that was made maybe weeks, months before is still active, right? And so you can imagine this ability to sequester these things and protect them and then release them as regulatory mechanisms. There are also mechanisms where we can shed them in exosomes. So the cell has a lot of machinery to regulate how much of that mRNA is both available for translation, either through degradation, production, or sequestration. Okay? All right, let's make sure that okay, so how degradation is going to occur is that you get the deadenylation of that poly A tail. Oh, this is like a full-on animation that's just going without me. I did not realize that. So you're going to get this this deadenylation of the poly A tail. These poly A binding proteins are going to release from it. So that's that, right? When these poly A binding proteins are now released from the molecule, this decapping protein now has access to that 5' end. It'll decap it, which then makes the 2 ends extremely sensitive to these exoribonucleases, and you get degradation, very quick and rapid degradation of that mRNA. The reason that you get polyadenylation, polyadenylation or deadenylation, is because unlike deoxyribonucleic acid, ribonucleic acids are much more sensitive to things like hydrolysis. Okay, so just simple hydrolysis via water will occur in RNAs where they don't in DNA. Okay, so the last topic before we close out the lecture today is going to be this idea of splicing. Okay, wrong direction. All right, so we talked about how in eukaryotic systems we have a single gene may have multiple exons. So those are the regions of that gene that are going to be expressed, right, into protein or RNA. And these regions of non-coding DNA known as introns. And you can imagine evolutionarily this idea of having modules, exon 1 being a module, exon 2 being a module, exon 3 being a module on a single gene, and instead regulating whether I put 1, 2, and 3 together, or 1 and 3 together, or 2 and 3 together. I now get a number of proteins with various modular functionalities from a single gene. And so the example that I will give is a protein that we study known as fibronectin. It's a very large extracellular matrix protein. It is a single gene that generates up to 20, we know of 20 isoforms of the protein. Okay? And it's all through splice variants. And certain modules, literally modules, get inserted during certain disease processes, and certain modules get deleted during certain disease processes, as well as developmental processes. And so this one gene has this plethora of proteins that come out from it. All right, so we talked about this idea of pre-mRNA. So we're going to get the RNA polymerase. It's going to synthesize what we call a pre-mRNA. It's going to have those exons and introns in that sequence. And now we're going to process that mRNA, so now it's mature RNA for translation. So the introns need to be removed from the primary sequence. And again, we're not going to go through a ton of the details because they get really complicated really fast. One thing I want to note before I move on is that the numbering scheme, when you look up, let's say you want to look for the PAD2 protein sequence or the coding sequence, the mRNA sequence, so you can transcribe or translate what that would be as a protein. When you look at something like GenBank through NCBI, what you're going to get is one of two things. Usually either an mRNA sequence or what's known as a cDNA sequence, right, which is a coding DNA sequence. And what that means is it's the sequence of the spliced mRNA variant, not the parent. Okay? So the numbering scheme will go 1 through 146 in this particular mRNA. And if you look at the convention, if we were to look at the pre-mRNA, you see it goes from 1 to 30. We don't count the bases in the intron. Picks up with 31 to 104. Again, we don't count the bases in the intron. 105 to 146, okay? All right. One way you can tell if it's mRNA versus cDNA, the DNA sequence of what the RNA transcript would be, is whether it's got T's or U's, right? Okay. All right, so how's this going to happen? Splicing of introns from mRNA occur at short, again, conserved sequences known as splice sites, which specify the beginning and ending of introns. Okay? A lot of research is still going on in this area because it's not completely clear how all of this works, but we do know some. Those splice sites are two base sequences. It's going to be a G and a U on the 5' end of that intron, and it's going to be an A and then a G on the 3' end of that intron. Those are designators of introns. Now, as you can imagine, there's lots of places in many, even exons, that have a G, then a U, and then an A and the G. And so there are other sequences that are recognized that help what we call a spliceosome, this complex of many proteins that are going to come together and splice that intron out, that help them recognize where the start and end of these introns are. Okay, again, catalyzed by a structure called a spliceosome. If you see these omics, it means usually a macromolecular complex with a very specific function. In cell matrix interactions, we call them adhesomes sometimes, or focal adhesions. Here it's called a spliceosome. And it's composed of various proteins and RNAs in what are known as SNRPs. And these are small nuclear ribonucleic particles, not proteins, particles. It's an easy thing to get hooked up on. So these are particles because they have RNA components and protein components that form these structures. Okay, so how the splicing reaction occurs. Again, here's our exon. Here's the 5' end of that first intron with the GU, the 3' with the AG, and we have this additional identifier, which is known as the branch point sequence, and is an A. We're not going to get into the details of what all these proteins are, but I want you to know conceptually what the reaction looks like, which is that we have a recognition of the splice site on the 5' end by this protein U1. We have recognition of the branch point sequence by this protein U2. And they're going to facilitate the bringing together of that RNA strand where the GU is proximal to that branch point sequence, that A. I will note that this cartoon is incomplete. If you're looking at it very carefully, the exon should extend, this dark brown area should extend all the way to that G. So just make a note of that. We haven't lost part of the exon. Okay? All right, and what's going to happen then is that this complex is going to facilitate a cleavage of that phosphodiester bond, and then that free end, that G and the U, are going to attack, again facilitated through this complex, attack this adenosine and form a chemical structure between those. So we're going to get a chemical bond occurring between those two. And this forms this sort of end loop structure known as a lariat structure. It looks like a cowboy's lariat, right? All right, and then this free end of exon 1, the 3' end of exon 1, is going to be facilitated to attack this site here, cleave the bond, and be spliced to it. So then we're going to have exon 1 and exon 2 together, and you get this lariat structure, which is then very rapidly degraded because the ends aren't protected, right? Or the end. There's only a single end right there. Okay? You can see I glossed over a lot of details. We obviously do not need you to know all of it, but again, understand this basic process. Okay? All right. Any questions on that at that very high level, screaming across the top surface of splicing? Okay. All right, that does it for us for today. On Thursday, we'll take the quiz, and then we'll talk about regulation of transcription.